Great. So as Darwin pointed out, so right from the beginning of the study of evolution by natural selection, this phenomenon, us, has been a target of inquiry. And as Darwin pointed out, the adaptation of man, or let's say humans now, perhaps in this day and age, is that we have reasoning powers in excess. Instead of definite instincts, this is a replacement in mental machinery, so analogous to what we see in bodily adaptation that it's not staggering. It's a target for the study of evolution, to understand how mental faculties evolve, just as we can understand how bodily adaptations evolve. So to take a kind of analogy, this is kind of what we want to be able to do for the primate radiation, for mental behavior. This is the Galapagos finches, and the adaptive radiation and the toolkit that they use is not inside their skull, but bolted to their face, the beak. The beak of the finch is what has changed as they've radiated to inhabit this island habitat.
Sorry, I'm afraid the mic isn't on yet. Oh, it is.
Sorry. It is on, sorry. If I stand a bit closer, it's better. I promised I wouldn't move, and then I moved, sorry. So what you can see, I think, here is that to understand the evolution of a natural feature in this way, we're not only looking at what problems the beaks have adapted to solve, so what kind of diet these birds are exploiting, but we can also see, I think, from this diagram how the form of that solution is constrained or enabled by evolutionary history. And this is the power of studying the evolutionary legacy, studying humans as another primate and not just on their own, is that we can see that the format of our thinking may owe a lot to that evolutionary legacy, whatever it may turn out to be. So here is here's a different view, if you like, on our relatives. This is a wonderful paper that has to-scale scans of primate brains. And we're obviously up in the top left, the largest brain in the primate radiation, but it's kind of color-coded here. So we can see that the other apes at the top there all have really large brains. In the kind of middle band, we've got the platyrrhine and the catarrhine monkeys with sort of intermediate-sized brains, and then the lemurs, the lorises, the galagos with much smaller brains. So we have information about the anatomy. We can see that there's a wonderful amount of variation for us to get a handle on, but what we don't really know just yet is what problems are these different minds solving, what kind of cognitive abilities are underpinned by these neural systems. So what I'd like to start by doing is looking at what evidence there is that indeed there are differences in cognitive ability across primates that go hand in hand with these differences in brain size. And then the question that I'll really be trying to grapple with for most of the talk is what differs? Can we narrow down what differs by taking a psychometric approach to this question? And the bulk of the studies I'll share have to do with trying to unpick the structure of executive function in chimpanzees and also in human children. I've got a little bit to say about direct comparisons. That's always riskier when you want to make direct comparisons between children and chimpanzees. And then finally, we'll talk about some work trying to model the trajectory of the evolution of different facets of cognitive control, and some virtual reality work with monkeys. Okay, so the first bit of evidence that there are indeed differences across primates that are consistent comes from these meta-analyses by Dina and colleagues that took the work that had been done with a variety of different primate species over the decades and sort of the 20th century on tasks that are largely to do with learning. Classic tasks like reversal learning, some tool use tasks, pattern string tasks, object discrimination learning sets. And they found that performance on these tasks correlates. So they called this a kind of G for primates, a general intelligence, because you could kind of get this correlation. And they are also related to the size of the brain, which, as I've explained, means it's related to phylogeny as well. So the larger brained apes performing well across these tasks, the smaller brained lemurs performing less well consistently across all of these tasks. But to say that there are differences in learning ability or problem-solving ability still leaves very much open what cognitively differs. When an animal is encountering a generalist animal like a primate rather than an animal like a finch that is definitely going to find a certain type of food like seeds to eat, then there's a lot that they could learn. The first handle, if you like, that evolution has on shaping the response to that environment could be what's attended to. So there might be some perceptual, attentional biases to attend, for example, to social stimuli. So I don't need to say that in this room, but that's a very useful way to canalize learning to useful information. There may be input analyzers that make that problem, some automatic fast analysis to get you started on some of these important events. And another important bottleneck is going to be the window of attention, how much you can attend to and sustain that attention in order to learn relationships. Another set of levers, if you like, that evolution could play with would be what is learned? How is the information represented? How long is it stored in memory? Are there world models that are being built that can be used productively and generatively to generalize? How abstract is that information? And then, as we heard beautifully in the first lecture from Lisa Feigenson, this can then feed down into what you attend to, what information you seek. So if you have expectations now or prior information about how the world should behave, then violations to those expectations may well have you seeking further information, and perhaps we get the start of things like metacognition curiosity. But once that happens, the challenge is for the agent to arbitrate between that top-down information and the information coming in from the environment. So you may need to control your attention between an internal and an external locus, inhibit prepotent responses that may be more driven by the stimulus, and keep in mind this goal in order to maintain behavior, for example, to seek information. So for many people, this seems like an obvious place to start if we want to try and narrow down what differs perhaps between primates, because this could be a real bottleneck on how much learning and adaptations to do with learning can end up influencing behavior and being useful. So we were involved in one of these studies that said, well, let's actually go ahead and test this idea that there are measurable differences between primates more directly. So rather than meta-analysis, targeted experimentation with two tests of inhibitory control. The top test that you're seeing in the video is the A not B error, so that's a nice familiar one for a developmental conference, where something is hidden in one cup repeatedly, the animals recover it, and then it's moved to another in the next trial to see if they can inhibit that perseverative response. And the second is a detour reaching task, where after some experience that you should, with an opaque tube that you need to take it to go around to reach a reward, you're then confronted with a transparent tube, which could elicit a prepotent response. You need to take the detour nevertheless. And so what we found, this is just the primate data here, but what you can see hopefully quite clearly is that performance, which is kind of depicted by the size of these circles, is very good in the great apes. It's intermediate in the monkeys. It's weaker in the lemurs. So there was a correlation with whole brain size. There's a clear phylogenetic signal. Indeed, it does seem like there are differences just in these simple tests across the primate radiation in inhibitory control. The second large-scale study that we were involved in was conducted by the Many Primates Consortium. And Manuel Bon is here, and I have to take the opportunity to call him out because he is really a fantastic instigator of this fantastic collaboration. The living lynx capuchins, the Edinburgh monkeys, are in the top middle there, looking at their cups. And this is a really simple test where a piece of food is hidden under one of three cups and in full view of the monkey, and all they need to do is remember for 0 seconds, or as close to 0 as you can humanly manage, 15 seconds or 30 seconds. And we look at how they perform across a rather small number of trials. So here what you can see is, from a later paper sort of reanalyzing these data, again, a really clear phylogenetic signal. So the apes performing well, the monkeys intermediately, the lemurs less well, and a relationship, again, with brain size. So it seems as if, again, there are measurable differences in this time, short-term memory, across the different primate species. So we're making progress, right? Well, it's still quite difficult, I think, to know. So stuff is differing, that much we know, but still to pinpoint what differs remains a little bit elusive from these kinds of data. So this has been pointed out by comparative psychologists, probably going through a midlife crisis or something for a long time, in the 60s and in the 80s, that when we compare species with a single test, and we see this one is doing better than this one, and we design that test to measure cognition. So they differ in cognition, right? Well, maybe not. Maybe they differ in their motivation, their level of experience with the test materials, something else that you haven't been able to completely control for. And one concern in particular with the A not B error task is that intensively studied species may be more used to tracking human hands, seeing a human hand as predictive of something important to attend to. And Sarah Gelbert and colleagues actually found that with the birds that they tested, the crows, that if they gave them a little bit of training just on the tracking of hands, their performance on that inhibitory control test, which they hadn't been trained on, improved. So experience obviously matters, and we know that. But it can have rather radical effects on performance. In the case of the short-term memory test, I think we have 3 measures per species, and we can see actually equivalent performance when there's no delay. So at least we know that animals are motivated, they can respond above chance, and then we're introducing this memory delay. So I think we're doing a little bit better there, but it still leaves open, have we correctly diagnosed the difference? Is this a specific difference in memory, or is there a more general difference that can be indexed by this test? It could be indexed by many others. So does this really matter? I mean, we've argued that it does matter, that if we want to reconstruct evolution, there's lots of interesting questions we can ask if we are able to correctly diagnose cognitive ability more precisely, such as, are there trade-offs between abilities? So, or is there correlated evolution? You can get good at memory, but it has to come at the expense of your attentional control, something like this. And these could really help us to elucidate the path, particularly that humans have come down. So it's trying to tell the difference between, to show it kind of more imagistically, the case in which we hope is not the case, there are no real cognitive differences, there are just differences in experience on the far left. So we know we've got differences in ability from our lemurs, our monkeys, our apes, but what makes up those bars of difference is harder to tell from these studies so far. I think with the short-term memory test, we can be more confident that there is a cognitive difference, but what exactly underlies that is less clear. Okay, so hopefully you're feeling a little bit depressed now, that maybe this is a really difficult problem that we are not going to be able to solve. And I'm going to be able to step in with our 3-step program that can help you through these times. And this is bringing in kind of the insights from psychometrics. So there's a huge source of variation that these studies don't yet harness, and that's individual variation. We're looking at variation across cohorts, across species, and that's fantastic. But if you've worked with these animals, you know there's extreme amounts of variation within these cohorts, just as there is with human children. And so that's an available resource for us to use in kind of individual differences analyses. So can we look at how these abilities hang together across individuals? If you're good at memory, are you also good at inhibitory control? And use factor analyses or some kind of latent analyses to see if we can unpick the structure underlying performance across multiple tests. So the first step here is to do a little bit more like was done with the short-term memory test, establish a signature rather than just doing a point measurement in the test of the different constructs, establish some kind of signature, hopefully at least have a baseline, so that the variation is more likely to be signal and less likely to be noise. So that's just to show what I was saying before, that our squirrel monkeys are performing okay, above chance, on the 0-second delay. They do really struggle once there's a delay. God love them. They're very cute, but they find this test difficult. But our capuchin monkeys are showing a sort of more linear decline with increasing delay, doing well at 0 seconds, but also they're above chance at 15 seconds. And I'm not showing you here the chimpanzees, but they're sort of over here. So they're doing well at all 3 delays. But I think for the point that I'm making here, this is what's really important, that they're above chance at the baseline. Okay, so then the next step is to to make sure that the variance that you have within these tests for the groups that the individuals that you want to compare is useful. So it's not at floor, it's not at ceiling, and it's consistent. And that's one of the trickier parts, because that means repeated testing, or at least enough trials that you can do kind of split half reliability. And here I again wanted to call out Manuel, because he has a wonderful paper taking this challenge really seriously and doing repeated tests of some of the most kind of popular, if you like, or well-known tests of primate cognition, and looking at that reliability. So some of them show good reliability over quite long periods of repeated testing. So you can ask him more about that if you're interested. And then the gold standard, or the goal, is to develop a multi-trait, multi-method test battery. So that's trying to get not just convergent reliability, so multiple tests of the same thing correlating, but also divergent reliability. So if we're testing multiple traits, then we should, if we think that there is a trait of, say, working memory, we want to see convergent reliability on that, but that it diverges from another test of another ability. And that's what we'd need to unpick the latent structure. That might not be there, but you want a test that can, in principle, diagnose that. So kind of one of the most famous multi-trait, multi-method test batteries for looking at executive function is Miyake and colleagues, Miyake et al. 2000, and subsequent work. And what they did was say, well, okay, under the umbrella of cognitive control or executive function, we have these three putative skills: working memory updating, shifting of attention, and inhibition of behavior. They have 3 tests per ability, and the participants do all 9 of these tests, and they have a large number of individuals to try to unpick the latent structure. So I won't go through all of these tests, but the memory tests are largely dealing with lists, keeping in mind a list and keeping it up to date in order to recover a piece of information. With the shifting tests, it's mostly rules like addition, subtraction that you're switching between. And the inhibition is mostly about stopping behavior in the face of a prepotent signal. So the structure that they uncovered in the second paper that is listed here showed that there was indeed correlation. So the idea that there's this umbrella term, executive function, seems to be grounded in reality. There really is a correlation across these tests. There's a unity to executive function that could be uncovered in this factor analysis. But then there was also diversity. There was discrete variance explained for the updating tasks and for the shifting tasks. Not so much for the inhibition tasks, which all loaded on to the common factor. Other models are available, but this was the model that we were reading at the time when we designed our test battery. You can sort of see where I'm going with this. So these were the authors that contributed to this work, and I particularly want to call out Christoph Folter and Eva Reindl, who were postdocs at the time, who helped to develop these different tests, and also tested a large number of primates and children on multiple tests in order to try to uncover the structure of executive functions, now in chimpanzees and in young children. Elisa Felscher also worked to test the chimpanzees when she joined later as a postdoc. So these were our design principles. We wanted tasks, funnily enough, that were non-verbal. We didn't want to have to train the chimpanzees to follow instructions. That means they need to be intuitive, intrinsically rewarding, ideally with ecological validity, so we don't need a lot of training. So there is lots of work within kind of the learning theory literature on different components of executive function in primates and rodents, but often these tasks require a very long training regimen and quite specialist equipment. That's what we wanted to avoid. The other principle we had was to try to cross-pollinate surface features and underlying construct demands. And that's because some reasonable criticism, I think, of the multi-trait, multi-method test battery is that not only the underlying constructs, but also some of the surface demands are quite similar within domains. And in fact, the one where the least similar, maybe the inhibition tasks, the stop signal, the Stroop test using language, the saccade using control of the eye movements, that's where you don't find any unique variance. So we explicitly tried to avoid that by having 3 roughly situations for testing using the materials of the comparative psychologists, cups, boxes, and things that people in a workshop knock together out of polycarbonate. And we have one of each in each construct, as far as is possible. So if you're a cup specialist, then you're not going to be able to see a structure just because we test all our working memory constructs with cups. We can debate how well we've achieved that, but that was the design principle. Sorry. And our participants were preschool children. Eva tested 185 preschool children before, unfortunately, the COVID pandemic put an end to our data collection around nurseries and schools in Northeast Fife and Edinburgh. And we worked with chimpanzees in 2 African sanctuaries in Kenya and Uganda by collaborating with our fantastic colleague Esther Herman. Okay. So I haven't said much about children. Maybe I'm a bit nervous to talk about children in this audience, but what we know for children is that when we're looking at the development of executive function, throughout actually all of childhood, all the way into adolescence, there is changes in capacity, so differences in ability in these kinds of skills. But in terms of that structure, it seems less clear that we see that diversity of ability. So unity, yes, but the latent structure is less clear. And mostly, factor analyses find support for a unidimensional model. It's quite, I don't think until we'd done this, there had been a full kind of 3x3 test battery. There is a sort of paucity of tests available for testing young kids that are not so verbal and not so keen on following rules. So we thought hopefully we can contribute to this. Maybe we'll find converging support for this kind of idea by working with kids. But obviously we wanted to do it in a way that allowed us to be comparative. So what I want to do next is talk about the task design work we did within each of these constructs, starting with inhibition. So we wanted to have these signatures, establish a signature for each of our constructs. And in the case of inhibition, we were again relying on the fact that when you see food, you reach for it, you go towards it. So we're using transparent materials. But we were checking that prepotent bias exists, so that this is an inhibition test, and making sure that that had been picked up on by our participants. So we did a study with the chimpanzees in Leipzig before we went over to test the chimps in the sanctuary, looking at this new test of inhibition, which made use of these small cups. Inside one of the cups is transparent, and that cup can't be opened, even by a chimpanzee. That takes some work, believe me. But these were well-sealed cups. And the other cups are opaque, and underneath you can find a peanut. So the task for the chimpanzee is not to lift the unopenable transparent cylinders. Instead, don't waste your time with those, just go straight for the opaque cylinders. It's a little bit difficult to see the video, but I think the action is happening in the right place. So of course, they first have to learn that these cylinders are not openable, but the prepotent bias is there. You're finding some rewards in the green cylinders, but not yet picking up on the fact that you shouldn't bother with these transparent cylinders. There's a bit of evidence that it's really quite annoying for the chimps. This nose-wise is a gesture of slight frustration. So that's what session 1 looks like. Quite a bit of the first lifts are directed to the transparent cylinders. But this actually surprised us how well they did on session 2. So first reach, it's an opaque cylinder. Next one, opaque cylinder. I wanted to kill the sound, but I just killed the video, sorry. So I think somewhere within the second session, you can even see the chimpanzee touches the transparent cylinder, but doesn't lift it. So he's overcome the prepotent response to interact with these transparent materials and is performing in quite a controlled way. So here's some data for that. So this is the first 12 lifts ever. And you can see that the initial lifts on the x-axis there are mostly towards the transparent cylinders. So they're not, on the y-axis is, are you doing the right thing? So they're doing the wrong thing initially when they're in the condition here where they need to, that I've just shown you, avoid the visible food. But there's a rapid improvement over these choices. And then reassuringly, the opposite is true if you need to lift the transparent cup to get the food. So if you have to do the obvious thing, then that's indeed what they do. So in this initial work to establish that signature, we can see that our inhibition test is posing an inhibition demand. Okay, so the other two tests follow that same logic and yield a similar signature that we can see evidence for this prepotent response. Attention shifting. So cognitive flexibility, as I said, in the adults, is really mostly measured through needing to administer two quite arbitrary verbal tasks, like performing addition, then performing subtraction. Not very amenable to comparative work. It's adapted for preschoolers using something like this card sorting test. So many of you may know this, but the task is to sort bidimensional cards in one of two ways. So you may start out in the shape game, in the color game, and here, okay, we're playing the color game, we're going to put the red cards on the red pile. And 3 to 5-year-olds can do that just fine. And then you go into the post-switch phase, and you're told, now we're playing the shape game. We're going to put rabbits on the rabbit pile and boats on the boat pile. And interestingly, the 3-year-olds just keep on perseverating and putting the red cards on the red target. So the targets are these two bidimensional cards. 5-year-olds follow the instructions, and so there's a developmental difference in this ability as measured by the DCCS. So we also wanted to make use of bidimensional materials, and there is a tradition of that in this intradimensional, extradimensional shift test that's used with rodents, for example, in drug development. And we piloted this, or we did a study of this with 95 preschoolers before we began the test battery. So the way this works in the imaginatively named tray, shifting tray task, is that we have 2 trays where we'll hide a sticker on one of them behind an occluder. And each tray contains filling material and an object like a cup or a box. One of the dimensions in those trays is irrelevant. So it might be that you need to work out the sticker's always in the red paper. But these are the chimpanzee materials here. But what you can see is that what varies over trials is the composition of what's in the tray. So it's not always the case that the green goes with the white paper. On the next trial, the green box may be sitting on the brown material. So your job is to ignore the irrelevant dimension and learn the relevant dimension over trials. And there's no instruction and no feedback. So that's different to the dimensional card sort test. Once they've learned to find the sticker, the children are split into 2 groups in this study. There's the intradimensional shift group and the extradimensional shift group. For the IDS group, everything stays reasonably the same. So we have new materials, but it's still about material. So they may need to learn, okay, stickers are in the blue wool, and keep ignoring the cups. But for the EDS group, they now need to ignore the material and pay attention to the cups. So they maybe, for example, can find stickers wherever the orange cup is sitting. And our hope is that that task imposes more of a cost than shifting within a dimension. That would show that attention is firstly captured by one dimension and then needs to be shifted away from it. So I'm going to show you a video of a chimpanzee because you didn't bring me here to show you videos of children doing this task. But there were children in this paper. Let me just, this is keeping you from falling asleep, I know, but it's probably a bit loud. So when you select a tray, you get the whole tray. So there's no real clue from the way that you make your choice which dimension is relevant. That's what you have to figure out over trials. The location can also vary, and of course, you can pay your attention also to the location. So it's quite a challenging task. This chimpanzee is doing quite well so far. This is the million-dollar question. It's very exciting. Will he get this one? Yes. So the predictive dimension is paper in this case. Okay. So we did get the result that we predicted. The extra dimensional shift is more difficult for children. They perform significantly worse when they need to shift their attention over dimensions. One thing that I thought was quite interesting is that we don't actually see any evidence of developmental change in the 3 to 5 age range in this test. And I thought I'd take a little side quest to talk about that briefly because of some of the discussions we've been having yesterday. So Buss and Spencer have written some papers showing that you can link performance on the dimensional card sort test to children's ability to use labels, for example, in a sorting test. And what they've suggested, and they have actually shown through some nice modeling work with a sort of dynamic model, is that these kind of labels can help to facilitate an all or nothing discrete shifting behavior. If you can pay attention to that label as you hear it in an instruction, this can help you make a discrete shift. Without it, because of the nature of this task, because 50% of the time, if you're on the wrong rule, you'll still be rewarded, it's quite a difficult task to learn. And so probabilistically, you're left with kind of drifting to one or the other and learning a little bit about both dimensions. Why shouldn't you? And so shifting is taking a little bit longer. In support of this notion, if you don't provide the instructions, if in the dimensional card sort all you get is feedback, that's right, that's wrong, and not, now we're playing the shape game. Don't forget, it's the shape game. You also don't see developmental change in the 3 to 5 age range, and actually kids find it quite difficult until they're 5 or 6, I believe. So there's an interesting side note to think about there about what's the engine of change for children once they have the ability to use these labels. Having said that, of course, we're not going to use the labels in the test battery, so we'll use the test where they are, at this point, performing pretty similarly across this age range. But if we went into a higher, up into 5 or 6 years of age, it may well be that self-directed speech would be doing something interesting and changing performance. So the last construct is working memory, and I'll share one of our tests that we did to design these working memory tests. So again, this was work led by Christoph Folter, and we were adapting a developmental study, the spin the pots task. So the task here, and all participants enjoyed this task, there's rewards everywhere. Your job is just to avoid redundant search, but you can have all the rewards. So you're searching in these cups, little boxes, until you find all the rewards, and we just measure how many redundant searches you make, and that's an indication that you failed to keep track of your past searches, which are self-directed, self-initiated. We might go through this left to right, and that's a really good aid memoir if the cups aren't moving, but once they start moving, so you take them away and you've just got to track by feature, you can't use that kind of heuristic anymore. The chimpanzees, as it turned out, didn't really use that heuristic, neither did the young children. So I'm showing you here performance on the task where the cups stay in the same location, and so they can use space and feature to avoid redundant search. And you can see that performance is really quite good in the chimpanzees. We're getting all the way up even to 6 boxes. In fact, it's fairly comparable. So the performance here means you make no redundant searches. So you search correctly without making any redundant searches. And one chimpanzee in particular was extremely good at this task and made no redundant searches, even with 6 boxes. It's a bit of a challenge for the experimenter, so I did feel for Christoph and Elisa and Eva to know what's going to happen, but yeah. The signature that we wanted to look at for our test battery was the effect of interference. So for working memory, the idea is that you're keeping this information live. There's some manner of rehearsal. Obviously, we don't think this is linguistic, but we might use a phonological loop of I've done blue, red, yellow. I'll do the pink one next. And you can elucidate whether or not there is that kind of active maintenance of information in working memory through introducing interference. And the more similar that interference to the information that you are trying to keep live, the greater the effect should be, theoretically. And this is what we found. So in this task, the chimpanzees are choosing on 2 platforms, interleaved. You make your first choice on platform 1, then you come over to platform 2, then you come back to platform 1. In the different boxes conditions, there's another array of boxes, but they have different perceptual features. In the food distraction condition, that you just come and get another bit of food and then go back. And then in what we thought would be the most challenging condition, the boxes are the exact same array. So even for us, that would be quite difficult. I've done pink, so I should avoid pink. Oh, no, not on this one. So you've got to keep the information separate and resist that interference. So we can see that effect of interference, and this is the signature we were looking for, that indeed the way that they're solving this task involves some active maintenance. Okay, so these are the 9 tasks, and this is the performance now of the individuals in the test battery, so just doing the test conditions, not some of the control conditions that I've shown you in the task development phase. And the question is, did we get good performance, usable variants? Did they participate? And we got really good participation, which owes a lot to the skill of the researchers doing the work, I have to say, especially with the children, to keep them going over all of these different tests. We had a lot of stickers going on at that time, so there were a lot of different categories of stickers that you could bring out to keep children motivated. And lots of lovely variation as well in the performance on these tests. So we will proceed to look at the results. We followed our 3-step program. We're on to step 3. And this is the confirmatory factor analysis. So there are some significant positive correlations across these tests. They're fairly low in the sort of their correlation value, but that is comparable to kind of like the Miyake et al. Test battery. What about the factors, the structures that we're trying to confirm or disconfirm? So 3 different models were significantly different to the baseline where nothing is related to anything else, the 9-factor baseline model. The one-factor model, the one common factor, which is the one that we might have most predicted, but given the literature to date. But we couldn't say that that fit better than either a 3-factor model with 3 different factors, or one of the other structures that we included where working memory and attention shifting load on one factor and inhibition loads on another. For the chimpanzees, we had to use exploratory factor analysis, so we did exploratory factor analysis for both chimpanzees and children. Testing 55 chimpanzees on 9 tasks is no small amount of data, but apparently it's not enough data for confirmatory factor analysis. But still, we can have a look at what latent structures emerge bottom-up rather than being imposed top-down. For the children, we get a reasonably similar story. So I'm sorry, it's a little bit small, but the best fitting model was, again, a 1-factor model. And you can see the loadings of the different tasks on that factor. But very close second was a 2-factor model where you have most tasks loading on 1 factor and just one of the inhibition tasks on another. For the chimpanzees, the story is quite similar. So a 1-factor model fit best, but a very close second was a 2-factor model where the inhibitory tasks, which are at the top, load on 1 factor, but attention shifting and working memory both load on a common factor. The overall amount of variance explained is reasonably small, sort of 17, 18%. There's plenty of variation that we're not accounting for with these models, but nevertheless, they are significant. So conclusions. We're seeing support for the unity component. Performance on these tests correlates. If you're good at one, you're good at another. If you're bad at one, you're bad at another. That's really interesting for us to know. And this seems to be the case in the children as well, to the best of our ability to detect it. But we don't have decisive evidence for the diversity component, and for that, it's because we can't diagnose specifically what kind of structure might exist. So one possibility, and this is an emerging field within the human cognitive literature as well, is that we just don't yet really have the right theoretical models to explain the different components. There are separable components, but we haven't quite tested the right theoretical latent structure. And I think that's really interesting. So there's lots to think about there. So it's possible that really the difference is how you make use of the interplay between your knowledge, beliefs, motivations in different contexts, and their context-specific variation would be very large, which is true. There are other ways to carve up this space. So one that Aurélien Frick and his colleague Nick Chevalier have put forward, which I think is really interesting, has more to do with whether control is self-initiated or reactive. So conditions are changing and I'm able to adapt versus a self-directed mode. And this is a little bit related to the DCCS. So older children perhaps using their own internal labels to help them control behavior. Of course, our sample sizes are not, it was Herculean effort to get them, but they're not enormous. So it may just be that we could do better with a bigger sample. But the main take home that I wanted to get is that I think from the best of what we can say with the work that we've done, these things are all connected. And I think from an evolutionary point of view, that's fascinating. If you have adaptive change in one of these skills, paying attention for a bit longer, controlling attention, or having a larger capacity working memory, other things are changing as well, is what this data would suggest. Now, that can be because there's underlying commonality in the mechanisms, or it could be that there are correlations over evolution, that these abilities evolve together because there is a benefit to one that facilitates adaptive change in another. So I'm going to explain that more in the next section. I wanted to talk a little bit about, well, you tested chimpanzees and kids on the same tasks. Were there any differences in the measures you used? Two of our measures gave us really nice signatures. So one of them was this shifting shelf task, where on one shelf you have to choose in the silver cup on the bottom layer, and on the other shelf you have to choose the brown cup on the top layer. So we teach them this first, and they show us they can do it by shifting back and forth between the platforms without making a mistake for 6 choices. Then we add in distractors. These are neutral distractors. You should never choose them. You've never chosen them. They are quite well able to resist falling for those distractors, and they keep moving back and forth and making good choices. So that stage doesn't take very long. And then we hit them with the test, where, as you maybe could guess, the same cups are on the top shelf, but you're on the left shelf, so you should ignore them. So this chimpanzee has correctly chosen his silver cup. He's done that 3 times. He's going to shift over to the other shelf. The previous day with the neutral distractors, this was no problem. I'm on this shelf, it's the top I should be using. I need the brown cup. But there's that silver cup. I'm giving the game away, but that's going to capture his attention. Yeah. Elise is very disappointed in him. And the next choice, he shifts away. So you can see that in this data here, both for kids and for chimpanzees, that when they make a mistake, it's to that exact distractor that was right on the other side, but it's wrong over here. So nevertheless, they're quite good at this test. It is difficult, but they are able to resist that capturing of attention and do quite well shifting between these 2 tests, these 2 shelves. And I'm showing you now the children broken down by year, their year of age. We didn't get as many 5-year-olds as we might have liked. That's because of the COVID pandemic. Children stay in preschool until they are 5. So at the end of that calendar year, you get more 5-year-olds. At the end of that calendar year, we weren't testing anyone. But we have enough children to see a significant difference. 5-year-olds are significantly better at that test than younger children, and only 5-year-olds are significantly better than chimpanzees. So chimpanzees and 3- and 4-year-olds are both controlling their attention in comparable ways in this test. This is one of our working memory tests. Instead of redundant search, this was not as popular. This test, you have to remember where it is. So you see where it's hidden in one of 4 boxes on one array, and as in the other test, we're moving between the 2 arrays to make our choices. I've been talking for a long time, so I wonder how many people can remember where that first one went. He's chosen, is he going to choose? He's choosing box number 1, is he right? Well done. Yes, he is. So here, we're still analyzing this data. Eva is busy building her own human, so we have still some work to do to process this data. But if you just plot their performance, you can see again this pattern that there is developmental improvement in the working memory. In this case, both 4 and 5-year-olds are outperforming chimpanzees. All of them are above chance. So it's not the case that chimpanzees can't do this test, but their performance is overlapping with 3-year-olds. So it looks like some differences are emerging, but quite late in the preschool years, possibly working memory emerging before control of attention. Remember that there's no evidence that these are completely discrete abilities from the factor analysis, but potentially these 2 levers at the same time, development and comparative psychology, are giving us more leverage to pull these things apart. It would be wonderful to test more species, of course, and more age groups, because this would give us more information, for sure. Okay, so to summarize this section, the phylogenetic comparisons that we're making in a cognitive way can really benefit, we think, from our 3-step program. And there are correlations between tasks involving working memory and attention, although the latent structure still remains a little bit elusive. There are differences in cognitive control performance, we could say, between chimpanzees and children from about the age of 4 or 5. And such differences could have really large downstream consequences in a developmental cascade for the sorts of abilities that we've been talking about in the last 3 days that also seem to be changing at the age of about 4 to 5. Things like learning sequences, modal thinking, reflective reasoning. Depending on the test, we're seeing some of these things changing at around this point as well. So, thank you. I'm good for time, I think. Oh, sorry. I'm so sorry. I have 10 more minutes, and I'm gonna use them. I'm really sorry. That was optimistic there. But I haven't got much left to say. So I want to talk about the work we're doing currently next. So there won't be lots of results here. But what I wanted to talk about is the fact that while in comparative psychology, and certainly in my lab, we've been focusing on these different kinds of skills, trying to understand what they look like in other species, trying to find the best ways to measure them. I think a lot of the action is in the relationship between the skills over evolution. And that's a thorny problem. And so we need friends. I needed friends to try to tackle the relationship between these skills. So I've been working with some colleagues in Princeton in the lab of Tom Griffiths. And this is work funded by the TWCF. He and his postdocs, Evan and Cameron, have been developing computational models of working memory and looking at evolutionary models about how different components of working memory could co-evolve. And in St. Andrews, myself, Andrea Miskov, who's here, who's hopefully you saw her poster, and Emma McEwen, are trying to make this an empirically tractable problem. So we need to work with them from the beginning to make sure that it's going to map onto something that we could eventually do with primates, and we are running some studies with primates. The central premise here is that for a natural system, investment in one cognitive ability is going to come at a cost to others in principle. We have a limited amount of resource. We should be rational and efficient in how we deploy it. So it stands to reason that there are going to be trade-offs and relationships over evolution between different kinds of cognitive ability. The model that we're looking at was developed by Sucho and Griffiths for working memory. And it's looking at, let's say we've got to remember 3 objects. We have this theoretical construct of quanta. Now, these might be discrete bits, they might be probabilistic, but there's an amount of resource we have available to remember these 3 things, and then they're gone. And so over time steps in the retention interval, as we're trying to remember these 3 objects, we can lose information in the system of quanta as one turns into another. This is what happens in the model. So it's a kind of drift model. Now, if I give you a cue and say, that's what I'm going to ask you about, then over subsequent time steps in the delay, if you're able to, you could allocate more attention to that representation. And so that's what the model can do. It can use this parameter control to control where the quanta goes. So they don't drift at random, they can drift towards the thing that you want to remember. If you want to know how that works with maths, please ask Tom Griffiths. I can't tell you, but that's what it's achieving in the model. So we have these two parameters, the number of quanta that you start with, that's something like your capacity, and this parameter of control, and that is the degree to which it resists random noise. So it can be very strongly resisting noise, or there can still be quite a lot of noise, but it's helping a little bit. In the end, the probability that you remember the information you wanted is directly related to your number of quanta, but not linearly. Okay, so I'm going to go very quickly through this. This is the work that's been done by mainly Cameron, looking at modeling the evolution of these two parameters, capacity and control. On the x-axis, you have the benefit for memory. So if you're in an environment where there's lots to remember and lots is changing, the idea is that there's more benefit, metabolic benefit for memory. And on the y-axis, we have how much you invest in these two parameters. So as benefit for memory begins to come up in the model, initially, this favors capacity. Control can't do much if you don't have much capacity. So first you build your capacity. But once you have some capacity and the benefits go up more, suddenly control comes into action. And the effects of the two synergize. Once you have a bit of control, capacity is actually much more useful because you can use it so much more effectively. And so that relationship between the two, the synergy, non-monotonic synergy is really interesting. Once you get into very high benefit and very high investment, it's back to capacity in the model being the most efficient way to get more bang for your buck. Control, you just need some of it, enough of it, in the way that the model works. Now, I know I'm glossing a lot of details here. This is more of an advert for a forthcoming paper, and if you're interested in this, this paper should be out early next year. When we're going to try and test these abilities, we're going to develop kind of retro cue paradigms where we can ask memory problems and give prompts and cues to see how much those can benefit monkeys. One of the new ways that we want to test monkeys has to do with a virtual environment. So I promised there'd be a virtual environment. So this is the last thing I wanted to share with you. This is Andrea's work, and she's implementing that task I showed you right at the beginning with the 3 cups, the short-term memory task. But now you're looking at 3 hedges from an elevated viewpoint in the virtual environment. And if you can approach the hedge that contained the food, then you're rewarded with rewards of that type. So this system was developed by our colleagues in St. Andrews, Josep Paul and others. And Andrea is the first one to implement it with the capuchins in Edinburgh. So this is a harder trial. So you can see that now the food has been occluded. The location of the food has been occluded for, funnily enough, about 15 seconds. But of course, we're showing you our star little monkey, Maturi, who's very good at this test. The rest of the time, the monkeys live here in the Living Link Center to Human Evolution. If you're in Edinburgh, I very much encourage you to visit the zoo and visit our monkeys. The fun part about the data that you get from this kind of study is that it has a time course. So here you see monkeys starting on the hill and heading to the 3 different destinations. The blue traces are monkeys who are correct. The red traces are monkeys who ended up in the wrong place. And what I want you to notice about this is that some of them started out wrong. So that's almost an error of encoding, could be a failure to attend. Some of them start out in the right direction and get lost halfway along, as if they've lost the memory trace. There's one really interesting observation that Andrea had, which is that sometimes monkeys turn back and go back up the hill. So it's almost like they've gone, oh, where am I going? I'm going to go back and check. We don't know for sure if that's what they're doing. It's an isolated observation, but that's what Andrea is going to try and find out in her next experiment. So watch this space. Okay. Finally, in evolution and development, I think these interactions are to be expected, these synergistic interactions between different components. When you change one part of the system, it has ramifications across the whole, and it makes a change to the adaptive landscape, also to the way that the whole system can function. This might be how you're feeling right now, by the way, is how many studies? And I hope that through these kinds of cross-disciplinary collaborations that we're engaged in now, we can start to grapple with this complexity. I think that complexity is the norm. We're not going to have many answers where, hey, you just tweak this one module and you get a human. I think that the problem is going to be complex, and there's going to be lots of complex interactions. So hopefully this will allow us to start to uncover the answer, if the answer is that what differs is everything, everywhere, all at once. Thank you again very much for your attention. It's really wonderful to be here in Budapest. We've been enjoying this conference for many years. Here we are. It's tonight, the gala dinner, that we're very much looking forward to. A couple of short plugs. If you want to see more monkeys playing computer games, please follow our newsletter. We do a monthly newsletter, and you can see lots of fun videos that our coordinator, Kate Grounds, puts together each month. And if you like interdisciplinary collaboration, and you're here, so I guess you do, we have a new center in St. Andrews where we're trying to do some capacity building and network building for people interested in diverse intelligences. And so you could join our mailing list. We have a funding call just now for if you're interested in organizing a workshop, an interdisciplinary workshop. We have got some funding to distribute that we were trusted with from TWCF, for good or otherwise. And that's, yeah, thank you very, very much to these people, and thanks again.
Okay, question time. Giovanni.
Hello, yeah, thank you, that was wonderful. Could I quickly ask you if you could speculate just a little bit on the kind of what you think could be the adaptive problems that are, you know, that could have selected for these differences? Because, as you're saying, you know, they might be correlated or they might be individually selected for, but we seem to be quite significantly different from them. So.
Great question. I guess it's a whole other talk, right? So that's the ultimate perspective. What causes these changes, especially in humans, is probably going to be just as multifactorial as the question of mechanism, I think. I think that there probably isn't a silver bullet. I think it's maybe a combination of different factors, a perfect storm, if you like. The apes are all generalists. They all do extractive foraging. They're all highly social. Loads of the monkeys are as well. So within the primate radiation, it's quite difficult to isolate different factors that could have shaped intelligence more than any other. Maybe this is a chance for me to plug the work of convergent evolution. So you can look in the radiation of birds. You can look in the radiation of reptiles. And really look at a broader diversity of life to see how cognition changes. So we have very large brain birds like the corvids and the parrots. So evolution has run this experiment, it seems, more than once. Perhaps we get more leverage by looking to other radiations to see if there are common answers. Having said that, I'm not so sure that there will be common answers. They may be different, may be diverse for a reason because they're responding to different challenges. So I guess if I have a speculation, I would say I think it's a perfect storm, a combination of different factors for humans, but definitely things that have been written about collaboration, a higher reliance on social information and culture seem very compelling as well.
Thank you.
Hi, thank you. Really interesting talk. I was just wondering if you had any thoughts about the purity of the tasks you used. For example, the inhibitory control task with the transparent and opaque cups. It seems that the participants may also have to have remembered that the opaque cups contained a reward, and also that they've tried the transparent cups before and they didn't open.
Yeah, absolutely. So the task impurity problem is really at the heart of the motivation for developing a multi-trait, multi-method test battery. I don't think we'll ever get pure tasks. This one, as you rightly point out, involves some memory to remember what, and some learning. It's actually kind of worse than that, because we did some further experiments with the Leipzig chimps, and you notice they were actually very good at that task. So they fell for it for a bit, but then they're like, you know what? No, I'm not picking up that horrible cup again. That learning was actually very rapid. We were quite surprised by how good they were. If you just give them red and blue cups, and there's food under the blue cups, it takes them a long time to work out, just don't touch the red cups. They don't do that nearly so well. So there was something about how those cups were sealed, we think, that helped them to use, kind of scaffold their performance from their object knowledge. This is no longer a container. I don't like things that aren't containers. When I'm looking for containers, I'm not going to touch this again. So we did a follow-up where we sealed the red cup and the blue cup was open, and the chimpanzees learned much quicker to avoid the blue cup in that case. So even sort of core knowledge can come in. And if you want to compare species, maybe they don't all have that core knowledge. So I guess my response to your question is no, they're not pure. And that's why you need this kind of multi-trait, multi-test, this big test battery to try to see if nevertheless there is a component that is inhibition. So we have the signature that makes it seem like it is inhibition. But their inhibition control is good in that case. It's very good, and it's bolstered by the fact that they kind of had a reason to avoid the cups, I think. Doesn't make life easier, but yeah, that's...
Anyone else from the back? No? There are more in it?
Thanks for that. That was really lovely. I had a question about the brains, because I was thinking that if you could, for example, look at the correlations or the decorrelations between brain areas, particularly in frontal areas, maybe you could get a sense for how much you'd expect the task to be similar or different. So I was thinking about the work of Leah Krubitzer, for example, where similar-sized mammals have different areas that they subtend in their cortex for somatosensory cortex. So I was wondering if the task might be auditory for a bat versus visual for a comparable species. Would you be able to see more of the, would they be more comparable in that sense, and would you find greater evidence for some commonality or difference?
Great question. I think that's so important. And just as we want to get more detailed and more fine-grained on the cognitive side, surely becoming more detailed and more fine-grained on the brain end, not just looking at the size of the whole brain, but actually looking at connectivity, looking at brain areas, is going to allow us much more of a handle on this question. I think if it does turn out that there is some dissociability in the cognitive side, then there ought to be corresponding dissociability that we can look at, and that would be really helpful to know and to see if we can predict one from the other. Ultimately, we should be able to, right? Yeah, absolutely. It's beyond my expertise to do the work, so I hope that there will be more collaborations with neuroscience in the future. I'll have to ask you for that reference.
Any more questions?
I'm just wondering how the things that you find relate to some higher level differences between humans and non-humans, for example, or different primate species, and whether you would maybe suspect that these kinds of findings could also influence how we test for other species differences. So for example, if you manage to find a task, for example, that takes into account your findings, whether that could also illuminate that maybe some other differences are not really due to real differences, but just in how things are tested. I don't know if that makes sense.
Yeah, thank you for the question. I would definitely advocate for more plurality in investigating. First, you have to understand what you're measuring. You have to look at capability. So it makes sense that first the tests should be narrow. But then once you start to uncover differences, I think it is right to want to broaden out and see what really causes that difference also in development. There might be some kind of, I guess one of the things we'd like to know is what's developmentally prior, what is a necessary change that can have a downstream consequence versus what is a de novo difference in and of itself. So yeah, I think so, but I'm kind of sorry to say it because it's quite hard.
Lovely talk. You started with the Darwin finches and the different beaks, and the story that tells us is that these are adaptations to the ecological niche. So if you want to see differences in cognitive faculties, maybe you also want to look at cross-species comparisons where the differences in the ecological niche are as big as possible.
Yeah.
So in terms of group sizes, how they feed, is that something you're thinking about?
Absolutely. I mean, there's kind of two things. When you're making choices about which species to compare, you also kind of want to maximize phylogenetic distance, so you're not kind of pulling from a narrow pool. But then another principle could be, well, actually, let's go for closely related species that live in a very different environment. It just so happens that capuchin and squirrel monkeys are quite closely related and have very different dietary niches, but they live in exactly the same bit of forest. I mean, the capuchins are literally upstairs. They live in the higher part of the canopy. So the capuchins are really extractive foragers, real generalists, and the squirrel monkeys are more reliant on insects, flowers, things that they don't really need to extract. And they take the manna from heaven. They take what the capuchins deliver. So those are the two species we have. And in other work, we found really interesting differences, really striking differences between the cognitive performance of those two monkey species, and they're both catarrhine monkeys. So I think that that's a really good idea. The problem is everyone has a different pet theory for which one is the most important thing. So if I say, well, I'm going to look at the dietary niche, someone else might say, but why? They're both social monkeys. They're really similar social structure. What a waste of time. But I think it's a good idea that we should be principled in choosing which species to compare to get the most leverage possible. At the moment, I've only really done chimps and humans on this work. But in our many primates, it's something that we do discuss. When we have an opportunity, often it's pragmatics. You've got gibbons, great. Let's test those gibbons. But if someone says, well, I'm at a zoo and they've got 4 different groups of primates, which one do you want me to put my effort into? Then these are some of the things that we're discussing. To try and get a broad phylogenetic coverage has tended to be the most important thing, but I think this is a really important point as well.
Okay, Susan.
Thanks for a glorious talk. So I have a question about your common factor. You're thinking of it as a common executive function factor, but you have no basis for doing that because everything is correlated.
That's right.
So what do you think that common factor, do you have any speculation, unless you have diversity of that factor with something else, you don't really have a factor of what is? So how are you thinking about that common factor?
Great question. I thought question time was over, actually. That is absolutely fair comment. Potentially, with those inhibition tasks, if we had a bigger sample, perhaps that would be our diversity. So attention and working memory hang together, not the inhibition tasks. But you're right, we need to go broader. So if we really want to show its unity here, then we need that divergent validity somewhere else. And that's always a possibility when you do 3 things. You should have done 5. The hindsight bias. You have to start somewhere. So I can't, in all honesty, say that I've got any right to speculate as to what the common factor is, not having given that talk. Said that no one else had the right either. I would like to think that there is something there in the ability to control attention and be goal-directed. That's my hunch, that if we did do that work to look for the divergent validity, we could find it. But I think better than that would be to find a theoretical structure with constructs where we could get tighter sort of diversity. I feel like that's probably there. It's just it wasn't this structure. So it's one way or the other, and both of them are enormous amounts of work. So I'm hoping, I think I need to spend a little bit of time getting feedback and reading and thinking about what the next step should be. But yeah, the point is well taken, definitely.
Anyone else?
Yeah, sorry, maybe I missed this point, but did you test also young chimpanzees? And so, okay, but do you expect the same kind of trajectories that you found in young children with like 5-year-old being better than 3-year-old in some tasks?
Great question. No, you really don't want to find young chimpanzees in these sanctuaries because they've been removed from the pet trade. So I'm happy to say there weren't that many young chimpanzees when we were there to test for us to look at development. It will take a really big coordinated effort to do that kind of work, and maybe it would make more sense to do it in zoo house populations where there's controlled breeding over a longer period of time. I think so, yes, I think there would be, but then again, some of the most interesting differences are occurring at this really intriguing time for the children, 4 to 5, where maybe what we're seeing is the, apropos a lot of the discussions we've been having, the role of language. They're learning language, they have this whole new symbol system they can use, and they're making use of it, even without explicit instruction. So it would be a really interesting case if it's like, well, the human developmental trajectory has this step function, but the primate developmental trajectory doesn't really, like a 1 or 2-year-old chimp is already doing about as well as they're going to do. Usually if we see age effects, they're kind of a U-shape. So the sort of adolescents are performing the best. There is some change, like the younger ones are not as good, and then sadly for me, as I approach middle age, the older ones are facing cognitive decline. But nothing, I wouldn't predict something quite so step-like, but again, that's just speculation.
Okay, last question.
Thank you. I actually had wonderful results. I actually had a related question to the very previous one. And I was wondering whether, because now we are comparing a developing system with a fully developed cognitive system, right?
Yeah.
So I was wondering whether there is, of course, comparing to young chimpanzees is very difficult for various reasons, but I was wondering whether it would be useful to have some baseline similarities, or to make sure that the differences don't come from simple greater ability for statistical learning, for instance, or a difference in general learning abilities, or a difference in how they update their models, or anything which could be characteristic. You see what I mean? But maybe you have implemented such baselines which would say that in general learning they are the same, but then they are not the same in this specific inhibitory component.
Yeah, I mean, that's a great confluence of kind of 3 of the questions that we've already been discussing. Can we get that divergent validity? Is there such a thing as a pure learning task? Would we be able to look at that developmentally? I've come to think there is no such thing as a pure learning task, that, whenever you pose a learning task, you're always bringing, there's a lot of parameters that shape how quickly you learn, and every one of those tasks is different, and they are going to be different to different individuals based on their recent and more distant history with those components. Unless you're working with 6-day-old rats, and you can really control that very tightly, it's really, really difficult to do that comparison when it's about learning, where out of its nature, if you're learning, we hope you learned something before, and you're going to bring that to the table. So I think probably the way that I've set it up is, well, all of these things feed into learning, but can we look at, through kind of this method, the role of different components on that learning ability? So I wouldn't predict that I'd be able to neatly isolate that out and say that. So I think your point is well taken, that with the kids, as they're changing, what changes in those kids to mean the 5-year-olds are better? I've just shoved something out there and said maybe it's because they learn language, but it could absolutely be all sorts of different things. And because I don't have a discrete factor in that factor analysis, I can't say it's their working memory. Look, I've been able to do it in a test battery, and that's the score that's different, and these other ones are not. No, it could be anything, but it's something. But pinning down exactly what it is, I'm not sure. From work we've done, looking at how they deal with objects, and basic problem solving in that age group, 3 to 5, and chimpanzees haven't seen big differences. When they're older, yes, but not big differences in that age range. But yeah, I'm not sure. Sorry.
All right. Thank you for the great talk, Amanda.